Hello, good morning, everyone. Thank you for being on time. We're going to get started. We will wrap up lesson 3 today on cells and move on to lecture 4. Um, Probably not going to be able to finish lecture 4, though. We'll see how much we covered. And whatever we don't finish, we'll finish it on Wednesday. And I'm just going to give you a little bit of update about the upcoming quiz, which is next Monday, I believe. And I'll do that when more people show up. Um, we are almost finishing the first half of the course. So, there's going to be a test coming up in about 2 weeks' time as well. So I'll give you more information on that when we have more people joining. Last class, we were talking about different kinds of organelles that you will find inside a cell, and we discussed the endomembrane system as well as other type of organelles that are not part of the endomembrane system. We talk about the mitochondria, for example, which is found in both animal cells and plant cells, and their job is to produce energy using glucose and oxygen as reactants. And in the process, you will create carbon dioxide as well as water. Those are the ingredient for photosynthesis. And photosynthesis happens in plants, but not exclusively in plants. There are other organisms that are capable of doing photosynthesis. Right? There are these single cell organisms that lives in ponds sometimes that can do photosynthesis. And they would have these organelles called the chloroplast. Chloroplast contains chlorophyll, which will take the carbon dioxide and water, capturing the sunlight and using that energy to produce glucose and oxygen. In the plant cell, for example, they would then take the glucose and use it in their mitochondria to produce energy, right, in the process of cellular respiration. So mitochondria are found in both animal cells and plant cells, whereas the chloroplast is only found in plant cells, but not animal cells. So that's sort of where we left off last time. We're going to look at a few other things that are kind of unique to plant that are found in plant cells, but not in animal cells. So over here, we have what's called a central vacuole. A central vacuole usually takes up a lot of space within the cell. Right here, I would say 80 to 90% of the cell is made up of the central vacuole in this particular case. And the function of the central vacuole is to store water as well as other nutrients for the plant cell. Okay, and in some cases, depending on which part of the plant you're taking the cell from, right? If you're taking it from the petals of the flowers, then the central vacuole is a place to store the pigments. The pigments is what gives the flower color. And in some cases, if the plant is poisonous, then the central vacuole can be a location for storing those poisons as well. Okay, so water, nutrients, sometimes pigment, sometimes poison. We do not have central vacuoles. Our animal cells, we don't have that. It's only found in plant. Now, there is something called a contractile vacuole, which is different from a central vacuole. Central vacuoles is found in a plant cell. Contractile vacuole is found in these single cell organisms, usually pond-dwelling, freshwater dwelling single cell organism. This one you might remember, we saw this fellow earlier in the lecture. This is a paramecium. Okay, and it's a single cell organism, and it lives in fresh water. And one of the challenges of living in fresh water environment is that water are constantly coming into the cell, which will cause the cell to swell up and burst. We're going to learn more about why that is the case in lecture 4. But the waters keep on coming in. It's like a boat with a hole in it, right? Water keeps on coming in. If you don't get the water back out, boat's gonna sink. In the same manner, if the paramecium don't pump the water out, the cell will swell up, and then it will burst and die. And that's what the central vacuoles are for. Okay, they squeeze the water out so that you maintain a normal cell size and don't burst and don't die. Okay, so these are contractile vacuoles. They're pumps, okay, that excrete water that are coming into the organism. In addition to the cell wall, cell membrane, which is a common feature of all cells, right? Regardless if it's animal cells, plant cells, bacterial cells, even the paramecium, all of them has a cell membrane, right? Also known as a plasma membrane, right? But for plants, they have an additional layer of protection outside of the cell membrane, and that is the cell wall. Cell wall is not selectively permeable. Okay? Things can just pass through it without much regulation, and they are made up of cellular fibers. Right? You learn about cellulose in lecture 2, remember? It's a type of polysaccharide. Okay? It forms the fibers of a plant, and the fibers of a plant are found in the cell wall of the plant cells. They protect the plant from bursting. So plant have a similar situation as the paramecium. They don't necessarily are like surrounded by water, but we do water our plants quite often, right? Like in nature as well, they soak up a lot of water from the environment. All those water cause the cell to swell up, and in order to not burst, they have a cell wall around it to prevent that from happening. It's like trying to inflate a balloon that is inside a mug, right? So the mug, oh, my mug's not showing. I'm trying to show you my mug. The mug serves as a protection, right? To prevent the balloon from bursting, and that's exactly what it looks like in the case of a cell wall. Okay, this is poll question. Why don't you read that, and we will launch the poll in a second. Remember, there are multiple answers here, so make sure you choose all of them. Okay, just a couple more seconds. All right, so in this question, we are looking for all the false statements, the ones that are incorrect. Right, so let's take a look. Smooth ER is part of the endomembrane system. So that actually is a true statement. The endomembrane system consists of the nuclear envelope, the rough and the smooth endoplasmic reticulum, Golgi body, as well as the lysosome, right? So that is correct, and therefore not what we are looking for. We're looking for the false statement. Next one, a lot of people chose this. Let's see. There is an abundance of mitochondria in muscle cells. Mitochondria is needed for energy production, right? Energy production. Your muscle cells move a lot, right? Your muscle moves a lot, so the muscle cell also needs to have a lot of energy in order to produce those movements. So it's not surprising to have a lot of mitochondria in muscle cells. So that is a true statement. We're not going to choose that. We're looking for the wrong ones. All cells have cell walls to protect itself from environment. That is false. Human cells, animal cells, for example, do not have cell walls. Bacteria, they have cell wall. Plant cells, they have cell wall, but not human cells. Hydrogen peroxides are found in lysosome. That is also false. It's not found in lysosome. It's found in peroxisome, right? Do you guys remember last time we had let me see which slide it was on so I can show you. It was on a page, slide 43. Okay, slide 43. We learned about, we learned about the peroxisome. Maybe I'll just go back to show you right here. All right, so peroxisome is where oxidation reaction occurs. That's like ripping electrons from other molecules, and it releases H2O2 as a byproduct, right? That's what the name means, right? Peroxisome, as in like peroxide. These are like circular sacs that has peroxides in it, okay? So that's why this is the false statement. It's peroxisome. Okay. Chlorophylls are found in chloroplasts. That is correct. We just learned that. Well, actually, we learned last class as well. So the correct answers, the ones that are false, are C and D. Does anybody have any question about this? So there were a few of these multi-select questions on the test, right? And I think overall it was done okay. Like it, you know, people generally did quite well with those. What was not done so well are the written response. Okay? I think people didn't read the question carefully. And for example, one of the questions asked you to write a hypothesis for the scenario where you're trying to see if driving with your windows open or closed is going to affect your fuel efficiency, right? So I think a lot of people did not read the scenario, and then they just write like their own hypothesis for like some other thing, right? So people say, you know, taking vitamin C is going to prevent a cold, right? So that's what I'm asking, right? That's the question. Did just say write any hypothesis, right? So you have to read it carefully and make sure that you understand what is being asked. Okay, class average was 72, so that's pretty decent. It's around the normal class average for the first quiz. And like I said, you have another one coming up on Monday, next Monday, that is, and that will be on lecture 3 and lecture 4. So cells as well as membrane transport. Okay, so I'll post the information on eCentennial later on, but it's very similar to your first quiz. There will be multiple choice questions, multi-select questions. There will be some written response as well. Okay. Next, we're going to learn about the cytoskeleton.
Sorry, Professor.
Yeah, go
For
ahead, please.
the test, can we do we have like a whole day to do the test, or is gonna happen in the class?
The quiz or test?
Test.
Test one? Yeah, test
Yes,
one
that's
is going to happen in a couple of weeks. Yeah, it's the same thing. It will be available from 6 a.m. to 11:59 p.m. And then you do it, whenever during that time frame. Okay.
thank
But
you.
again, right, once you start, you have to finish it, right? Okay. All right, cytoskeleton. Right. So cytoskeleton refers to a network of fibers that are found throughout your cytoplasm. Okay, so this is your cell. If we're talking about a eukaryotic cell, you would have a nucleus, and this is the cytoplasm, right? Cytoplasm. The cytoplasm is not just liquid, okay? There are a lot of these fibers, which we'll look at right now, and they all have various functions, right? They might support the cell shape, they might help things move around. All these fibers, they are called cytoskeleton. There are three main types of fibers that we're going to be looking at, and they're all listed here. First one is called microfilaments. Next is intermediate filaments. And then we have the microtubules. And, that's the cartoon depiction of what they look like. And we're going to examine each one of these in closer details right now, starting with microfilaments. Microfilaments are actually made up of two strands of actin proteins. Right? So proteins are, of course, a polymer. And in this case, we have these actin molecules that are linked together to form the actin strand. And then if you take two of them and then you braid them together, you would get yourself a microfilament. Now, this stuff is found throughout the cell. This here is a cell, and the cell membrane is it's like roughly there. You can actually use paint, I guess they're called fluorochromes, which would give off color when you expose the microscope slide to UV light, for example, and you can make the different components of the cell give off different colors. So in this case, we use antibodies to tag the actin proteins, and we make it so that it's going to show up as green when we shine UV light at it. And then the red here in the center, that's what's showing you the chromosome, so you know where the nucleus is. So all these green fibers, they are basically the microfilaments. So they provide this scaffold, almost like a net, that allows the organelles to attach to it, right? We don't want the nucleus to be like sloshing back and forth inside the cell. We want it to be kind of somewhat anchored, right, within the cell. And same thing with all the other organelles, the endoplasmic reticulum, the Golgi body, all that stuff should have a place to attach onto, right? That's what the microfilaments are there for. They provide attachment sites for various organelles. Also, it allows for cell movements. Do I have a picture of that? Oh, I don't. But basically, cells, most cells, they stay put, they stay where they are. But a lot of cells needs to move from one place to another within the body. They have to move around between your tissue cells, for example, and white cells does that. And in order to move around, these microfilament would rearrange. For example, all the microfilaments here on the left could break down and reposition themselves on the right side. And when that happens, the cell can extend forward on the right side, right? And then it would like grab onto something else and then pull the rest of the cell body with it. It's kind of hard for me to show you on the drawing, but like, imagine my hand as the cell, right? So like it would extend forward, right? It would grab onto something and then pull the rest forward, and then extend again and grab onto something and then pull the rest forward, right? So it's like this blob of slime that's trying to crawl forward. And in order to extend forward and make connection, we are going to need to have these microfilaments. Okay, so attachment sites for organelles allows the cell as a whole to move around in the body. That's the job of microfilaments. Next, we have these intermediate filaments. Intermediate filaments are important because they lock the cell membranes of two cells together, right? So let's say we have cell number 1 here, and then we have cell number 2 here, and we want to lock them together, right? What's allowing to glue them together is the intermediate filament. These things that allows you to lock the cells together, they are called intercellular. Between two cells, that's what intercellular means, junctions. Okay, so they are stapled together. And where in the body do you think we want to have these kind of tight connections between the cell? Anybody want to share some ideas? And why would we ever want to staple two cells together in the body? What's the point of that? Anyone? Tash said, so we don't fall apart. Yeah. That could be one of the reasons. But any organs in the body that you think is important to have a tight seal, right? So that things don't leak all over the place. Do you know?
Maybe everything.
Everything?
Yeah.
Yeah. Okay, but specifically, I was thinking about things like blood vessels, right? Okay, so you don't want the blood to be leaking between the cells and go all over the body. I'm thinking about intestinal tract. You don't want the food to be leaking out, right? So it's important to have these tight junctions between the cells. Okay, sometimes we do want things to pass through, right? Like, you know, when you're filtering your blood, right? We do want them to be able to leak out, and those are places where you don't need to have tight junctions. In addition to between cells, they are also important for providing structural support for some of the organelles. For example, the nuclear envelope is supported by a layer of a type of intermediate filaments called lamins. And what happens is when the lamin, which is an example of intermediate filament, which is a protein, is mutated, then that results in nuclear instability. Okay, it's going to cause your chromosome structure to fall apart, and that results in accelerated aging. Some of you might have heard of the disease called progeria. Progeria is a rare condition, but one that is very, very horrible and deadly as well. People with progeria has mutation in lamin A gene, and it caused them to age rapidly, right? So this person has all the feature of an old individual, but he's only about 8 or 9 years old, okay? And needless to say, they would die early on in age, right? Because their body is rapidly aging. There was actually a movie, this is kind of old, so some of you might not have heard of it before, but you know, Hollywood, they made a movie about this, although it's not an accurate depiction of the condition, but nevertheless, it's based on this condition. Julian asks, is it like the movie Benjamin Button? Yeah, Benjamin Button is backwards though, right? Which doesn't happen in real life. I think that movie was Brad Pitt was born really old, and then as he grows up, he turns younger and younger, right? So that's like the backwards of what is being described here, and something like that doesn't happen in real life. Yeah, go ahead, Tosh.
Sorry, I had a question about like something you said in a previous class. It's like not related to this. Can I ask you?
Yeah, sure, go ahead.
So you're talking about like trans fat, right?
Sure.
And you're saying trans fat is like the really bad one that like builds up in your body. You can't get rid of it.
Yep.
You said like Canada banned trans fat, but like
they did. Yep, a couple years ago, 3-4 years ago.
doesn't like margarine contain trans fat?
So like in the hydrogenation process, right, they might produce a little bit byproduct. I think what they really ban is the deliberate use of trans fat in food. So one of the one of the rationale of using trans fat is that it extends shelf life, right? So you know, they cannot have that in any of the packaged products now. And you cannot use it to fry the food at restaurant, right? To, you know, make it tastier. I'm not sure if margarine is obviously it's not banned, but I'm not sure if they have to like maybe remove it in the process or something like that. That one is it's an interesting question, but we're gonna have to look that up. Okay, maybe if you find out, you can also let us know. Because it's I do know you're correct, right? When we do the hydrogenation process, we do introduce like trans fat is a byproduct, but usually they would, you know, extract that and use it for something else. But I don't know, like if there is a minimal amount that is allowable, right, in these margarines and whatnot. But you know, something to look at.
Okay, thanks. Sorry, I was just curious.
No, that's fine. Okay, so that is intermediate filament. And you know, Progeria, because it's so rare, that you know, there's not a lot of funding that goes into it. But you know, they are breaking some grounds when they do in vivo, which means you do it directly in a living organism as opposed to ex vivo. They were able to reverse some of the syndrome in mouse model that has progeria. Yeah, so with the ability to edit genes and gene therapy, which you will learn in lecture 9, some of these diseases who was once incurable might have a chance. For 2 minutes, could you please update the Zoom link in the communication Zoom section, because many of us couldn't get through or we couldn't find the link to class. It's the same link that we have been using since the beginning, right? What do you mean? Which? So you want me to post it again? Is that what you want?
Yeah, maybe in the Zoom communication. I don't know if it's communication Zoom, because many of us couldn't find it.
It's the same one that we have been using. Here, I'll post it on the chat box. Okay, so you just save that on your desktop or something. I'll post it again, I guess. Okay.
Thank you.
Here, I'll do it now in case I forget. Okay, give me a second, guys. Now, the Zoom link is going to be different, right? When Professor Moran takes over. So, you know, this is just for my class. So I'll post an announcement now. Some students have trouble. All right, I just posted on eCentennial. It's gonna be under the news announcement. Okay, so please bookmark that and not use it. Do you guys know how to find the recording as well? Because I have been getting some emails for that as well. Let me see. Just give me a second. Copy link. Okay, so in the chat box, I posted the link. That's for the recordings, right? If you miss anything, if you join the class late, if you need to listen to things again, that's where you'll find it. Okay, anyone else have anything to say before we continue? Like any, I don't know, this doesn't have to be related to the lecture, like any housekeeping items? Yeah, go ahead, go ahead. Yeah, Tash, go ahead, please.
Sorry, I was wondering about the tests. You said they're not cumulative, like test 2 is not.
No. That's so test 1 is cumulative for everything I taught you. So lecture 1 to lecture 5. Okay, test 2 will be lecture 6 to 10. Right? So the individual tests are not like the final test is not for the whole course. That's basically what you need to know, right?
Okay, so the final test will be whatever we learned
From
at the
Professor
end.
Moran, that's right.
Okay, all right.
Yeah.
Thank you. And you said there's going to be a test 1 review on 12th of June?
Sure. Yeah. Whatever it says on the yep, 12th of June. That's right.
Okay, so we'll be going over like all the answers and everything
All the answers to the what?
for the test. Like
Uh,
what does the review mean?
so review, like we will do a test review. I will give you a review package, and we'll take it up. Yeah, basically, that's what it is. So like
Okay.
I'll go through like major concepts that you should know for the test, right? That's what I mean, right?
Okay. So sorry. So basically, like we'll be going over like what we got wrong and stuff, right?
No. We're not going to be going through the questions on the quizzes, right? So like I said, right, we don't return the quizzes because we have to maintain the integrity of the test bank. But for the test one review, right, we'll like I will give you a review package and then we'll take it up. Basically, that's what it is. Okay.
Okay. So I arrived a little bit late. I don't know if you already went over this, but like after the class, would you be able to, like, you don't have to give me the answers, but like let me know what specific concept I got wrong on the test, like afterwards?
So again, right,
Uh,
like that's not something we do because we have to. I know it's not ideal, right? Like people are, they don't like it when they don't get to see what they got wrong, but it's the structure of the online course, right? So I'm not able to share that information with you, unfortunately. Yeah.
Okay, so not even like just like not the question, but like just like the
No, I
specific
understand. Yeah,
concepts.
yeah. But like it's impossible for me to go through that for everybody, right? Like you understand, it's just not like practically
Ye-
speaking, it's not something that is feasible, right?
Okay, I understand. Thank
All
you.
right. Yeah. Anyone else have anything else they want to ask about the course? Just housekeeping task before I keep on going with the lecture so that we don't have to stop and address these things along the way and just focus on the lecture. It's okay if you have questions, but ask now. No? Everybody's good? All right. So the last one, the last cytoskeleton that you have to know is the microtubules. Okay, so microtubules are these long hollow tubes that again they're found, in the cytoskeleton and provide these tracks for vesicles to move along within the cell. Right, so like when we have a vesicle that comes out from the rough endoplasmic reticulum and it needs to get to the Golgi body, you cannot just rely on random movement for the vesicle to get there because it's going to take some time. Okay. And so there are these microtubules, tracks that are within the cytoplasm that allows the vesicle to get on it. And then there is a molecule that looks like a little person with two hands and two feet. That's actually what they look like. It will hold on to the vesicle, and then it will walk along, literally walk along these microtubules and get it to the Golgi body. Okay, so it provides some form of structure, right? So that movements are not randomized. They also provide structural support. Right? This is the growing butt of a neuron. It's trying to extend part of the cell to make a connection with another neuron cell. And to guide this extension, we need to have the microtubules. Microtubules can also be formed from other organelles that we have not learned yet. This one is called the centrioles, and the centrioles will send out these microtubules to help guide the movements of chromosomes when the cell is dividing. Okay, so these special type of microtubules, they are called spindle fibers. You don't normally find them in a regular cell. They would only form when the cell is dividing, when you're trying to do mitosis, right? Split the cell into two. And it's important to guide the movement of these chromosomes so that they know where to go, right? Remember, you have 46 chromosomes, and each of the daughter cells must get the same amount of chromosome at the end. So how do you know which chromosome goes where, and how do you make sure they don't get tangled up and move accordingly? That would be the job of the spindle fibers. Okay, so once again, we have the microfilaments, which help scaffold things, right? Help the cell move. We have the lamin, so the intermediate filaments, which helps in stapling cells together, right? Forming these junctions, as well as nuclear envelope stability, for example. And then we have these microtubules that help guide things within the cell, guide the movement of vesicles or guide the movement of chromosomes. Last two things that we're going to look at are these cilia. Okay, so cilia, earlier in the PowerPoint, I show you a slide of what they look like. Usually it's covering the entire cell, and they're usually useful for movement. The paramecium, for example, the single cell organism that I showed you with the pumps, they have cilia around them that helps them move in the fresh water. And within the body, we also have cilia that help move, not to move the cell around, but to move things that are near the cell. An example would be in the airway, in the trachea. If you have dust particles that are trapped, the cilia would move, and then it will move all the dust particles up to your throat, and then you can spit it out or you can swallow it. People who smoke, long-term smokers, they lose the cilia in the airway. And the only way to get those trapped particles out of the system is to cough it out. Okay, so they have what's called a smoker's cough, right? Any other idea of where you might find cilia in the body? In the female body specifically, males don't have that organ. So where in the female body do you expect to find cilia other than trachea? Both gender, of course, have trachea, but specifically for females.
In the reproductive system, the uterus or the fallopian tube?
The fallopian tube is correct, okay? Not quite a uterus, but the fallopian tube, for those of you who don't know, is the ovaries where the eggs are released, and from the ovary, it needs to travel to the uterus, right? So this is the picture of the fallopian tube, and these are the cilia. So there is the egg, right, that I just drew in, and the cilia kind of brush the egg along so that it knows where to go, right? Okay, to the uterus. All right, so that's another location of where you will find cilia. Okay. The other thing is a flagellum. Okay, flagellum is singular, flagella is plural. So
Okay.
usually, yeah, go ahead.
So would you say the cilia is like a vehicle?
They would help. Okay, so like they do one of two things. They either move the cell, such as the paramecium, or in the case of the fallopian tube, they are not moving the cell, right? The cell is right here. Now they are moving things that are around the cell. Okay, so they either produce movement of the cell or they help move something else along the way. That's probably the best way to think about it. Is that okay, Trish? The flagellum, which is either there's one or two per cell, and it produces these whip-like structures that help the cell to move. The only place where you will find flagellum in the human body is the sperm. Okay, and sperm only has one tail. Right? If a sperm has two tails, it doesn't go anywhere. It just swims around in circles. You can find that on YouTube if that's what you're interested to see. Some organisms, they do have two flagella. These are, again, a single cell pond-dwelling, freshwater dwelling organisms. It's possible to have two flagella per cell, just not in humans. Okay. And these are made up of microtubules, right? One of the three cytoskeleton that we talked about. And that's pretty much it, right? Just to refresh your memory, here are a list of things that are kind of unique to animal cells, but not in plant cells. You will find lysosomes, centrioles, and flagella in animal cells. In plant cells, you will find chloroplast, central vacuole, and cell wall in the plant cells. This is plant versus animal, okay? I'm not saying that you will not find chloroplast in other cells, right? In fact, I told you, right? I keep on saying freshwater single cell organism. They're actually called protists, okay? You don't have to know that, but you know, the whole group of them is called protists. And a lot of protists, they actually do have chloroplast in them as well. But I'm just saying, right? Comparing animal to plant one-to-one, you will only find chloroplast in plants, but not animal cells. Okay, this is just to see if you were listening to me. Which of the following is made of two intertwined actin protein strand? Okay, anyone else? Right, thank you for participating. The correct answer is microfilaments, right? That's the one that has two strands right here. You can see two intertwined actin proteins like that. All right. So that is that. Now, you know, I I actually don't know how you guys found the first quiz to be like. Was it difficult, easy? You know, was it like, you know, what you expected, or like it was completely totally different, right? I'm not sure. You can let me know if you want. You can let me know in the chat, or you can send me email to let me know. But what's really important, hopefully, after doing the first quiz, is that you do these study guide, right? Okay, so like fill it in yourself. I post the answer as well, right? And you can check your work against that. And you know, you can use these as references or as guide to help you progress through your evaluation, right? Your quizzes, right? So these are excellent summaries of things that you must know, right? You know, some people just print the answer key and use that. That is not a really good idea, right? You really have to internalize these things and be able to explain it in your own words, right? For them to be meaningful to you and to help you remember it. Okay, so please take your time to work through those. Yeah, some people say they have a hard time managing, but I look at the statistics because I can actually see how much time people take to finish it. Most of you were done well within the time limit. If you have trouble with time, maybe, you know, if you're stuck on a question, come back to it later on. And you know, don't dwell on a particular question too much, right? Like you have the option of just write down which question you're stuck on, and then on the left side you can skip directly to a particular question if you want, right? You don't have to keep on pressing back or forward to find that question. If you're stuck on question 14, move on, right? And then you just go directly back to question 14 when you need to. Yeah. Yeah, I mean, like, you know, if you prefer in person, right, we do have that option in our program as well. I actually do. I'm teaching second semester in person right now, currently, and then I have students who were online with me in the what was it, the winter semester, and then they switched to online, right? Switch back to in person. So there is that option. And I will post some information about online tutoring as well. You know, it's part of your tuition. Your tuition covers it, and they have tutor available either in person or online at the library. You won't be able to transfer it this semester, right? So like if you were in an online class right now, then you would have to finish the online. But then, you know, for next semester, when you choose your courses again, you would have the option of choosing, you know, the in-person option. You know, feel free to email me if you need more information on that. Okay, so that is
Professor,
sorry, go ahead. Yeah.
I think we cannot switch it to offline because when I called the college, they said if the first semester is online, you have to do a second online
Is that right? That's maybe they wait, did you call the college or you call our program?
program.
You call the program, okay. So maybe they changed the rule on that, but let me double check with the person, and I will update you guys on that. Okay. Yeah, they told you that as well. Oh, okay. Maybe they changed the rule around that. I haven't get any update on that. But you know, if you guys call, maybe that's the case. But I'll double check and update you guys on that when I get the information. Okay. All right. So let's do lecture 4. Lecture 4. Lecture 4 is about membrane transport. All right. So we already know there is a cell membrane that is selectively permeable. Okay. It hold on a second. It helps regulate, you know, what comes into the cell and what's leaving the cell. So we call that selectively permeable. And we know that the cell membrane is made up of mostly phospholipids, but there are other things that makes up the cell membrane as well. So let's just draw that out together, and you know, we're going to draw something like this, and hopefully it will make more sense when you draw it out. Okay, so this here is called the fluid mosaic model. Okay, so please take out a piece of paper. And copy it down. Fluid mosaic model. So let me just show you a quick picture of what mosaic means. You might have seen like a window like this. This is called mosaic window. Okay, a mosaic means the whole thing is made up of many different individual parts. So you got the blue parts, the red parts, and whatnot. And sometimes they form a picture, right? So these are called mosaic window. So the cell membrane, first of all, it's not a wall. Okay, if it's a wall, then nothing goes through, right? It's fluid, right? It means it's dynamic. Things moves around. And it's mosaic because it has different components to it. We know it has phospholipids in it, and we learned that in lecture 2, but there are other stuff in it as well, right? So let's try to draw this out together. Here is the cell, and here is the nucleus, right? And we're going to be taking a slice of the cell membrane, and we're going to see what it looks like. Okay. We're going to have these phospholipids. And you can draw a bunch of them. Some of them has kinks in their phosphor in their fatty acid tail, and some of them don't. Okay, so this is a phospholipid bilayer. And this would be the outside of the cell. And then this would be the cytoplasm, the inside. Okay. The outside of the cell is actually we call it the ECF. Okay. Is extracellular fluid. Right, outside of the cell. Now, along the way, you are going to have these big proteins that insert itself in the phospholipid bilayer. Right, I'm just going to draw more phospholipids here. Right. Now, I want to remind you on here maybe. Okay, so this is hydrophilic head. It's water loving. That's why it's facing the outside and the inside, mostly water. And then these are hydrophobic tails, right? If it's both straight, then these are saturated phospholipid. And then the other one with the kink, that's the unsaturated phospholipid. Okay. So more on that later. Now this protein that goes all the way through the cell membrane, that is an integral protein. Right? It passes through the entire phospholipid bilayer. Sometimes we have just proteins that are on one side and it doesn't go through. These are called peripheral protein. Okay, so integral goes all the way through, peripheral just on the side, either on the outside or on the inside. And they have different functions, which we will learn later on as well. Okay. Sometimes you would have, well, not sometimes, you will have some cholesterol here that are in the cell membrane. And the function of the cholesterol is to help maintain fluidity, okay, modulate membrane fluidity. Now listen for a second, right? You don't want a membrane to be too fluid. If it's too fluid, if it's too liquidy, things just leak in and out easily without any kind of regulation, right? So we don't want that. On the other hand, we don't want it to be too solid either. If it's too solid, then nothing goes through, right? So you need it to be just the right consistency, right? Just the right amount of fluidity. And cholesterol will help control that, to modulate, to regulate that, okay? So more cholesterol equals to more fluid. Less cholesterol will be less fluid. Okay. Any questions so far?
Or is that it? Like, is that like your little sketch complete?
Is my sketch complete?
Yeah.
No, we're still gonna draw more. Yeah.
Oh.
Don't take a picture yet. Yeah, if
Oh.
that's what you're asking.
Yeah.
So I have a question for you guys, since you don't have a question for me. If a membrane wants to be more fluid, if you want to increase the fluidity of a membrane, sure, you can have more cholesterol. But which one of these do you need more of? Do you need more saturated fat to make it more fluid, or do you need more unsaturated fat to make it more fluid?
Is it
Unsaturated.
saturated?
I think I heard both.
Unsaturated.
Yeah.
Unsaturated.
Okay, let's do a quick vote. My question was, if you want a membrane to be more fluid, do you need A, more saturated, or B, more unsaturated? Pick your choice. You want it to be more fluid. Do you need more saturated phospholipids or more unsaturated phospholipid? Choose something, guys. Even if you don't know, if you're just guessing, choose something. Don't leave a multiple choice blank, ever. Unless, you know, you lose marks for choosing the wrong answer. Okay, thank you. You know, we have like roughly half. To be more fluid, you actually need to have more unsaturated phospholipid. I'll write it down here. The unsaturated phospholipid make membrane more fluid. Why? Think about the saturated fat and unsaturated fat that you learned in lecture 2, right? Unsaturated fat is liquid at room temperature, right? Because of the kinks in the tail, the double bond that forms the kink makes it harder for them to pack together. If they don't pack together as much, they are more fluid. Okay, so unsaturated phospholipid makes membrane more fluid, whereas for saturated, it make membrane less fluid. Okay, we'll come back to this concept in a bit, but hopefully that makes sense to you. All right, what else do we have here? Sometimes you might have some sugar that are stuck on the protein. Right, these are carbohydrates. Okay, so if you have like sugary stuff that are stuck on the protein, we call them glycoprotein. Tell you more about that later. Sometimes you have sugar that are stuck on the phospholipids, so we call that glycolipid. Glycolipids. Okay. And on the inside of the cell, right, we talk about those cytoskeleton, right? The actin filament, for example, right? So they provide attachment sites for the peripheral proteins. Right? So the cell membrane is not going to be flopping around. Right? It's going to have some anchorage point. It's like, you know, building a tent. You need to anchor them to the floor. Otherwise, the wind will blow it away. So these are anchorage point that allows the membrane to stick. Okay, that's pretty much it for now. Take a picture if you need to. Do a screenshot. Whatever you need. Right? That's the complete picture. And again, if you have questions related to this, go ahead, Tosh.
So you said these notes, like specifically like these notes that you draw, do we have access to them?
Yeah, I post them afterwards, right? In online on eCentennial.
Okay,
Let me
I'll
show you.
check.
Yeah, here. So like if you go to the course page, content, I'll post here like class notes, right? That's what it says, right? So anytime you see the class, this is the stuff we took for lecture 3, right? So today I will give you the class notes for lecture 4 as well, right?
Okay, thanks.
Yep. All right, let's keep going. So these are the different components that we talked about, right? I told you what the phospholipid does. Basically, the whole, like I would say 90% of the cell membrane is made up of the phospholipids. You have the saturated and unsaturated version, depending on how fluid you need the membrane to be. And the kind of environment you are in will determine how fluid the membrane needs to be. For example, if it's a plant that lives in the desert, you would want the membrane fluidity to be different from one that lives in, you know, very cold environment. It's something to think about. We'll get back to that in a bit. Cholesterol will further help maintain the consistent fluidity. And then the glycoproteins or glycolipids, it's important for something called cell identity. In a nutshell, it helps your body to distinguish what belongs to the body and what does not, right? That, of course, is an important feature for your immune system. Right? So this is what I was talking about, right? How would the percent of saturated, unsaturated fat, and cholesterol content of the cell membrane found in plants growing in cold environment differ from those growing in hot environment? Okay, so basically, if a plant grows in a very cold places, do you expect it to have high or low saturated, unsaturated, and cholesterol content in the cell membrane? I'm gonna break you guys into group and give you like maybe 5 minutes to talk about it, and then we're gonna come back and see. Okay. Let me just. How many people we have here? We have 32 people. I'm just gonna do like 9 groups. Okay, 9 groups. Go for it. Okay, so again, your job is to discuss this question, right? Think about and rationalize what kind of content you need to have for a plant that's growing in a cold environment. 5 minutes, guys. Okay, welcome back, everyone. Yeah, I saw a lot of good discussion going on, a lot of mics moving. That's always a good sign. No, so thank you for participating. Does anybody want to share their discussion? What's the conclusion?
My group we came to the conclusion that the like the plant or the item that you're looking at is not going to, and please correct me guys if I'm wrong, but I think that it is not really manipulated by the environment that way to change.
Okay. If I understand your conclusion correctly, is that there's no difference between cold and hot environment in terms of saturated fat contents? Is that what you say?
So I think we were saying how like maybe in a drier environment,
Mhm.
the percentages would be different. And then we also discussed that basically the plant wouldn't change based on the environment. But I'm sorry if I maybe not wording it properly and I'm
No,
speaking
no,
for
that's fine.
everybody,
No,
but
no, that's okay.
basically that it's not completely manipulated by its environment.
I see. Okay, thank you. Thank you for sharing.
you.
Anyone else or another group maybe that they want to share the answer before I tell you what it is?
Well, I'm not sure exactly, but based on what we're discussing, well, some of us were going back and forth as to what we think, but we do believe that there is a change in plants
Mhm.
that grow in a cold environment than from that which grows in a hot environment.
Mhm.
I think we're thinking that if it's hot, then they probably do need more water.
Okay.
So then if they need more water, and they will be using more water, then it's probably more fluid, which will make the percentage of unsaturated fat increase and the cholesterol level will increase. Whilst on the other hand, if it's cold, then they probably don't need as much water. Yeah.
Okay. All right. I see what you're saying. Okay. Thank you for sharing as well. Okay. Good discussion. Good, conclusion. It doesn't matter if you get it right or wrong, right? Like it's about the process of, discussing, right? And, rationalizing, explaining why you think it is the way it is, right? But still, I'm going to tell you what the correct answer is. Let's talk about a cold environment. Okay. Cold environment. Okay, so if I look at the cell membrane from a plant that grows in northern Canada, okay, very cold all the time, okay, what would be the percentage be? If a plant is from a cold environment, the low temperature would cause the membrane to be more solid than it otherwise would, right? I need you to understand that statement. If it's really cold all the time, then the membrane have a tendency to be more solid than it otherwise would, right? Okay, like it's more likely to freeze up, right? So the plant does not want that to happen. It doesn't want the membrane to become more solid, because if it's so solid, so cold all the time, then nothing gets through, right? So as an adaptation to counteract that, the membrane wants to be more fluid. Okay, so cold temp makes membrane less fluid. So that's the first point. To counteract that, membrane needs to be more fluid. Okay, so how do you make the membrane more fluid? You need to have more unsaturated, less saturated, and then you need to have more cholesterol. Right? All these things would make the membrane more fluid, because the cold temperature is trying to make your membrane less fluid. Do you understand that? You want to counteract the change. Remember we talked about homeostasis, right? If it's really, really cold, in the environment and it's making the membrane solid, you need to counteract that. You need to make it less solid. You want to make it more fluid, and to do that, you need more unsaturated fat, less saturated fatty acids, and then more cholesterol.
That makes sense. If it's since it's connected to like homeostasis, as you were saying.
Right. Thank you. Kashina, you had a question? I saw you unmute. Oh,
No, I was the one that just
oh,
spoke.
sorry.
I'm sorry, it was me.
Okay, go ahead, please.
So is this kind of like when we go back to saying that maybe like it's not about it's about the population evolving. Does this kind of I'm layering over some things in my head and I just want to make sure I'm not doing that incorrectly.
Yeah, so like in I think I get what you're saying, right? So like this would be what has already happened, okay? But like, you know, over the course of evolution, to put that into that context, you would have plants that have cell membrane that have more cholesterol, and then some that have less cholesterol. Just focus on cholesterol for now to keep it simple, right? So those plants with less cholesterol in the membrane, they would probably like not thrive, not survive well in the cold environment, and they would die off, right? So those with more cholesterol would live on and, you know, produce more offsprings. And over time, the entire plant population in this cold environment would have high cholesterol content in the cell membrane. Therefore, they're more adapted to the environment. Does that answer
Yeah.
your question?
Absolutely. Thank you so much.
Yeah. So you should think about, right, the hot environment, right? And just to give you a few pointers, for hot environment, like things are more fluid now. They're more liquidy, right? Okay, liquidy, is that even a word? Probably, right? It's like more fluid now. So you need to counteract that fluidity. You need to make the membrane more solid. Okay, so please think this through. Okay, I'm gonna put like double star here, triple star. Think it through. Okay, nudge, nudge, wink, wink. Okay, think that through. Moving on. Now we will switch gear and sorry, I'm taking away your break today. We just have so much to talk about. I'm sorry. I hope you can, you know, bear with me here. Now, the next thing we're going to talk about is membrane protein function. We already know. Oh, you want me to go back to the previous slide? Sure. Here. You just yeah, maybe take a picture if you need to. Remember, the recordings are online, right? Okay, so if you need to hear the explanation of this again, you know, because it's triple starred, all right, you want to go to, let me try to do some calculation, maybe like 1 minute and 15 minutes. That's the mark that you're looking for, okay, in the recording that I will post after class. 1 minute and 1 hour and 15 minutes. That's when we were talking about this stuff, all right? Okay, so membrane protein function. We know there are these integral proteins that spans all the way. And then we have some of these peripheral proteins, which are just kind of attached on the side, right? So all these membrane proteins are very important. They have various functions. We're going to be looking at 6 of these functions, okay? They're all summarized here, but we will look at them individually. This is just to refresh your memory of the difference between an integral protein that goes all the way through from like a peripheral protein. Now, you might wonder, where do these proteins come from? Well, last class, we took a note. And you know, we make the proteins from the ribosome, from the rough ER. The vesicle will go to the Golgi body. It will get sorted, modified, and repackaged. And then some will become part of the cell membrane. So this vesicle with the protein in it, it will move all the way to the cell membrane, and it will insert itself there. And that's when it becomes either an integral protein or a peripheral protein. So what does it do? Well, we already kind of talked about this when we drew the picture, right? You see here, we had the cell membrane and we have some peripheral protein here that are anchoring the cell membrane to the cytoskeleton, right? And that's precisely what the function is, right? One of the functions anyway, one of the 6 functions that we'll talk about. It allows cell to attach to the extracellular matrix, right, the outside environment, but also to the cytoplasm as well, right, and cytoplasm. Well, actually, sorry, this is just referring to the cell alone, so it's fine to just say extracellular matrix. It helps maintain and support cell shape by providing anchorage points for actins and intermediate filaments in the cytoplasm. That's where I wanted to put it. Okay, so this is the red blood cell. You saw it in lecture 2 when we learned about sickle cell anemia, right? To maintain that very distinctive shape with the pinch in the center, you need to have these membrane proteins that helps to attach the membrane to the cytoskeleton. All these fibers that you see in this top picture, those are fibers in the extracellular matrix, and the cell needs to be able to attach to the sites, to navigate through it, to move around, to stay put in some cases. So that's the first function, attachment to cytoskeleton. Next, we have cell signaling. Okay, some membrane proteins are receptors, and they are responsible for transmitting signals to the inside of the cell. Right? A receptor has a key and lock relationship with its target. Okay, so this is the receptor. This is the outside, the ECF, extracellular fluid, and this is the cytoplasm. Okay, and these are the signaling molecules. Signal molecules. They could be a bunch of things, but as an example, we can say they are hormones. Okay, so they will bind to the receptor, which would then trigger a series of changes and reactions inside the cell. Right? And those changes would either change the behavior of the cell, or it would change the gene expression. It might turn on some genes, turn off some genes, and that would cause the cell to behave differently. Okay, so a lot of times the molecules don't go into the cell; they just bind to a receptor, and then it make all these changes. So an example here is adrenaline. Okay, so adrenaline is produced by there's something called the adrenal glands, which are on top of your kidneys. Okay, this is your kidney. Okay, on top of the kidneys, you have a gland. Right, so this is the adrenal gland. You don't have to know that. I'm just telling you in case you're interested in. You'll learn this in next semester, and that releases the adrenaline, and we need adrenaline for various things. Okay, fight or flight response when you're all pumped up and ready to run in a race, right? So that adrenaline will be released from an adrenal gland, and then it will affect different targets in the body. One of the targets would be the liver cell. Okay, your liver is on the top right side here, top right side here of your upper abdomen, and the adrenaline, which actually is the same word as epinephrine. You might have heard of epinephrine from EpiPen, for example. They are the exact same thing. Okay, just 2 names for the same thing. So it will bind to a target, and then it will cause a series of reactions which are collectively known as signal transduction pathway. Basically, it's a pathway that helps pass the signal on. And then in this case, the adrenaline will cause the hydrolysis of glycogen and release glucose for energy, which totally makes sense because you're getting ready for a race, right? Okay, so you need that extra glucose to be available in your body. So the adrenaline never went into the cell. It just binds to a receptor, and then it causes all these commotion inside the cell, which result in a response. All right. This always reminds me of my a story. Like when I was growing up, for some reason, like my family was like anti-Halloween. Okay, like they don't like kids coming around ringing doorbells and asking for candies. Like my mom was just like totally enough of that. So you know, every year when it's Halloween, you know, if someone rings the bell, my mom would be like, hide in the basement, okay, pretend no one's is home. I never really got that because now we already turned off all the lights, right? Us moving to the basement is not necessarily gonna, you know, make it do anything, but she likes to do that. She's like, you know, hide in the basement, okay, just don't make a sound, and then the kids will go away. So that's what we always do, okay. And you know, it relates to this because it, you know, the kids who are ringing the bell are like the adrenaline. They never came into the house, they never came into the cell, but they causes all these commotion to go on within the house, right? Okay, so it's kind of like that. So, you know, hopefully that story will help you remember this example. Now now that I have my own kids, I don't care. Like, people ring the bell, yeah, go hand out the candies, give them 4, give them 5, right? I'm glad, like, I'm not passing on my mom's anxiety when it comes to Halloween. So in another example here, we have what's called neurotransmitters. That's something that you will learn again in next semester, but that's how your neuron communicate with each other. We have one neuron here, and we have another one here. And if it wants to communicate, it needs to send out these neurotransmitters at the synapse. And the neurotransmitters would move to the next cell and bind to a receptor, and then it will cause the next neuron to respond. Okay, depending on what kind of neurotransmitter we have, the response will be different. Okay, you don't really have to like memorize this example. It's just like another illustration to show you this is like really happening in real life. Okay, neurotransmitter might sound like a big word, but I'll give you a few examples that you might have heard of, right? What do people call like the happy hormone? Okay, when you exercise or when you eat a cookie, it makes you feel good. What's released in your brain? What's that? Sorry?
Is it endomorphin?
Endorphin. Yeah, that's one of that's not the one I'm thinking about. That's what that one releases when you are like in pain, right? So it dampens the pain. Another one,
serotonin.
Serotonin is
dopamine.
also a neurotransmitter, but that one is for like regulate. It's like if you don't have enough of it, then you get depression. But this particular one, dopamine, thank you,
Dopamine.
Takira. Yeah, that's it. That's the one I was looking for. But you know, all the examples you guys talk about, the endorphin, the dopamine, the what was the other one? The serotonin. All these are neurotransmitters, and they all function by binding to a receptor, right? And then causing the cell to change its behavior. Okay. In case I lost you, we are talking about different functions of membrane proteins. First one is attachment to cytoskeleton. Second one is cellular signal, cell signaling. Okay. Next one is enzymatic activity. By now, you should know that enzymes are these proteins that helps speed up chemical reaction. The proper word is catalyze, right? Catalyze chemical reaction. They lower the energy required for reaction to take place. And sometimes, actually a lot of times, you need multiple reactions to be happening in order to get the product that you want. Okay, so here we have an example. We have a starting substrate. Substrate is the thing that the enzyme works on. It goes into enzyme 1, it becomes something else, but that's not the end of it. It needs to go into enzyme 2, and then so on and so forth. It needs to go through 4 different enzymes before you get the end product that you need, okay? And rather than having these enzymes kind of scattered throughout the cytoplasm, it makes more sense to put them right next to each other in the cell membrane. So when you finish with the first reaction, you can immediately put it in the next enzyme, and then you will be able to carry out the reaction rapidly, right? One after the other. So this increases efficiency, it increases the speed of the reaction so that you can get the end product much quicker, right? So these enzymes are embedded within the membrane, right? They don't have to be integral protein necessarily, it could be peripheral protein. The point is they belong to the same metabolic pathway. They belong to the same set of chemical reactions so that you can do them in succession very quickly. Next, we have intercellular junction. We already learned about this earlier, right? With the intermediate filaments. In addition to the intermediate filaments, you have additional proteins that would help glue the cells together. And it's important to have these junctions because we want to prevent uncontrolled movement of materials between cells. We definitely don't want things to be leaking out of our intestine. We don't want things to be leaking out in the blood vessels, right? So that's why we need to have these tight junction, intercellular junctions. Number 5 is cell-cell recognition. The glycoproteins and the glycolipids, the sugary proteins and the sugary lipids, they collectively form what's called like these are called like antigens, okay? But just put it in simple terms, we call them identification tags. Your identification tags, the kind of molecules that you have on your cells are unique, right? No one else has the exact same set of ID tags as your cell unless you have an identical twin. That's why when we do organ transplant, it's impossible to find someone with 100% match, right? You can find matches that are as close as possible to prevent rejection, but no two people are exactly the same, okay? There will always be some kind of difference. And so that's why people who receive organ transplant, they need to take immunosuppressants, right, to dampen their immune system a little bit, right? And it's the same thing with giving blood, exactly, right? You will learn that next semester. I actually just finished teaching about blood transfusion in my semester 2 last week, right? If you're type A blood, you can donate to type AB person, but not the other way around, right? So these type A blood, type B blood, they are essentially talking about the kind of molecules, the kind of glycoproteins or glycolipids that are found on the surface of your cell. Your immune system use these tags to distinguish self versus non-self, what belongs to the body and what does not, right? And you want to make that distinction so that you attack the right things. If your body doesn't recognize what belongs to the body and what does not, two things can happen. Number one, you don't know how to defend yourself from a bacteria. Okay? Bacteria, it doesn't belong to the body. If you don't see it as an enemy, you don't attack it, you're gonna get a major infection. People who are immunocompromised, they have that problem. The other potential problem is if you see a cell that belongs to your body, self, right, belong to yourself, and you thought it was foreign, and then you start attacking it, then that's a problem as well. It causes autoimmune diseases, diseases where your immune system turn against yourself. Does anybody know any example of autoimmune diseases that you read about on the news and social media? Lupus is an example of autoimmune disease. Very good. Maybe one more. Zomes, you had
Is it like MS?
Yeah, MS, which stands for multiple sclerosis. Not leukemia though. Okay, Trish. Leukemia is a mutation of the bones, blood cells. Sorry, one more.
Rheumatoid arthritis.
That's right. Yeah, that's right. Rheumatoid arthritis. When T cells attacks the cells in your joint. Okay. Okay, very good. A lot of excellent ideas. So that's what happens when you no longer recognize your cell as your own. All right.
May I ask you a super quick question just regarding the receptors in the last slide?
Sure, go for it.
I'm not sure if it's true. I've heard that receptors can stop working or not work as well with anything from medications all across the board. Is that something that's true, or is that?
Sorry, you were asking about this receptor. Is that what you're talking about?
Sorry, yeah. So when the receptors are taking in what the information or what so have you, I've heard that they can stop slow or maybe stop taking.
Yep.
I don't have the right words yet. I
No,
I
you're
think I will
absolutely right.
after
Yeah.
this.
So there are many conditions where the receptor is not doing its job, right? Maybe the signal molecule binds to it, but it doesn't transmit the signal as well, right? So,
Okay.
that could be a problem. So, for example, in serotonin, the case, right? The neurotransmitter might be released, but then, they might not be creating a response in the target cell, and that could lead to clinical depression, right? So,
Okay.
you would need to take medication that would prolong the effect of the serotonin. Let them stay in this is called the synapse. Okay, let them stay in the synapse for longer, so they can exert their effect for a longer time. Since we don't have enough action on its own, right? So that's one way we
Right.
can, fix that problem. But
Thank
you're
you so
absolutely
much.
correct. Yep.
Thank you.
Okay, so we again, I just love repeating myself. We talked about attachment to cytoskeleton, cell signaling, enzymatic activity, right? Cell recognition, forming junctions, right? That staple cells together. Those are the 5 functions of the membrane protein. And the last one, and the focus of lecture 4, is to participate in transport. Okay. So some membrane proteins are important for moving other molecules from one side of the cell membrane to the other side. Okay, and exactly how they do that and what are the different types of transport, that's the focus of the rest of the lecture. Okay, we still have like 50 slides for this lecture, but don't worry, we'll finish on Wednesday for sure. Now there is a multiple choice question here. And I just remember when I see this question, I just remember there is something I forgot to tell you. So you actually won't be able to do this, but that's okay. I'll take it up with you, and then you will understand. So according to the fluid mosaic model, which is the thing that we drew for our cell membrane, which of the following is a true statement about the membrane phospholipid? Okay. First, they are free, as in the phospholipids, right? They are free to depart from the membrane and dissolve in the surrounding solution. Now that is a big no-no, okay? We definitely don't want that to happen, right? Let's take a look at our picture again, right? What that statement was saying is, you might have this phospholipids who decided, no, I don't want to be the cell membrane anymore. I'm just gonna pop myself out, and I'm gonna bring my friend along, the one next to it, and then they're just gonna go into the surrounding and then dissolve and be gone, right? Think about that, huh? That's not a good thing. You're gonna have a random gap in the cell membrane, and if enough of it happens, it's gonna become super leaky, right? So that's not something that we want to see. That's why A is not correct. Number 2, or B, they, again, as in the phospholipids, occur in an uninterrupted bilayer with membrane protein restricted to the surface of the membrane. We know that's not true because we have integral proteins, right? Integral proteins are the ones that are able to span through the phospholipid bilayer. Okay, so that is not true, right? Because integral protein exist. And that's what you have to do when you see a multiple choice question on the test, right? Like you don't want to reject answer because if it looks wrong or feels wrong, right? You need to have a legitimate reason for rejecting it. And sometimes writing out what the reason is will help you with your guessing a little bit. Okay. Next, they have hydrophilic tails in the interior of the membrane. That's not true either. Okay, it's actually hydrophobic. Right, the tails are oriented to the inside of the cell membrane. Right, the hydrophobic heads are on the outside. They can move laterally along the plane of the membrane. That's the piece of information I forgot to tell you. Okay, and that is actually the correct one. Right, as the name implies, fluid. The membrane is not a wall. Okay, like on our wall, things don't move, right? The brick stays where they are, and for good reasons. But on a fluid mosaic model like this, let's say we color this particular one red, right? This one is okay. Like it can move around. It can like switch place with the one next to it, right? And then they could just move laterally from one spot to another. Okay. So think of I guess the best analogy to think about this is let me oh, the program just crashed. That's why it was doing that weird thing. Okay, so like the best way to think about this is I don't know if you can visualize this. Okay, I haven't really seen it done it in before, but like I can imagine this, right? Think of like filling your bathtub with water, okay? And then you dump a bunch of like ping pong balls in it or something, okay? Fill it all up with ping pong ball. The ball is gonna be bobbing around, right? It's not gonna stay in one place, right? It's gonna be like move around a little bit, right? That's exactly what's gonna be happening, okay? There are gonna be lateral movement from one spot to the other. What's really rare though, oh my goodness, what did I press? What's really rare is for the phospholipid to flip from the outside to the inside, right? Like if I take a look at this picture again, I said like if I paint this one red, it will move left and right, okay? But it's not gonna flip and go to the other side. Sometimes it could happen, but it's a very rare event, okay? It's not gonna do the flip flop, right? But it's able to move sideways, right? So the true statement is D, and everything else is incorrect. Okay. Now this one you can do. I'll launch the poll in a second. Okay, just a couple more seconds in case you want to choose something. So the correct answer is D, and most people got it right. Protection from lysis. That's the function of a cell wall, right? It has nothing to do with membrane proteins. Everything else is one of the 6 functions that we talked about. Okay, moving on. We're gonna spend the last 10 minutes talking about cellular energy, and then I think we'll stop there. So metabolism. Metabolism are basically all the reactions that are going on inside a cell. And there are like tons of reactions that are happening at any given moment, all right? Some reactions are considered as catabolism. Others are anabolism. So catabolism are reactions that involves breaking down molecules into smaller units, okay? And they tend to release energy in the process, right? So we for example take polymers, and we break it down into monomers, right? If you take proteins and you break it down into amino acid, that is an example of catabolism, okay? Or sometimes we can say catabolic reactions. Okay? And that releases energy. Energy. And it also releases a little bit of heat as well. Right? So we have daily experience of this, right? Especially in the winter. When you eat, you get warm, you warm up, right? Because when your body digests food, when your body metabolizes the food, it uses catabolic reaction, generates heat and energy. When we take the monomers and build polymers, there are many polymers we need to build. We need to build proteins so that we can, strengthen our muscles. We need to build proteins to make hormones. We need to build glycogen to store our extra glucose, right? All that building, right? That's anabolic reaction, right? Also known as anabolism. And that requires energy. Right? Think about building anything in life, right? Like, building a fence, that takes energy. But over time, the fence kind of falls apart on its own, right? And that doesn't require energy. That just happens spontaneously. Okay, so building things requires input of energy, and that energy is supplied by catabolic reaction. Okay, so the energy that was released in the catabolic reaction is what drives the anabolic reactions. Okay, that's why we have to keep eating to survive. We need to constantly generate energy to power these anabolic reactions. Okay, when your cell divides, it needs to make new DNA, right, from the nucleotides, right? That's anabolic reaction, and that requires energy. Right? Building anything in the body requires energy. So this is basically what I wrote down, but in a more cartoonish way to help you understand. These are food molecules, and the food molecules can be like carbohydrates, right? Protein, lipids, right? They are macromolecules. And in the catabolic pathway, we're going to break it down into building blocks. So maybe glucose, amino acids, right? Maybe fatty acids, right? And then we will either use it immediately, or we can build them into other useful molecules, right? Like glycogen to store them, right? Maybe we need to make hormones, right? So those are anabolic pathways. We are not very efficient, our body, okay? Like a lot of the energy that was that is in the food are actually wasted as heat. Well, when I say wasted, it's not really wasted, because I guess like we are warm-blooded, and we do need to maintain normal body temperature, but we are not converting 100% of the food into usable form of energy. Okay, think of like a light bulb, right? Like the really old one, the incandescent light bulb, the one with the filament in it, right? Those are not efficient, because if you touch it, it's like super hot, right? Okay, those are like less than 10% efficient. And most of the energy from the electricity is not converted into light. Most of it is converted into heat, right? So that's not efficient. Human body is similar. Okay, we are about like, I think 34, 35% efficient. Okay, the rest is converted into heat. So what about that usable form of energy? What exactly is that? That usable form of energy is called ATP. And I'll show you a picture of ATP later. And we generally produce ATP through cellular respiration. That is the main type of catabolic reaction in the body that allows us to produce energy. Right? And it's not the first time we talk about cellular respiration. Right? You take glucose and oxygen, and you produce water, carbon dioxide, and energy. What I did not tell you previously is that the energy that you produce is in the form of ATP. You know, part of cellular respiration happens in the mitochondria, right? Okay, and there are all these steps that are involved And we're going to learn that in lecture 5 next week. But for now, just know that cellular respiration is the main reaction that produces energy in the form of ATP. ATP stands for adenosine triphosphate. Adenosine as in like the adenine in DNA. It's connected to a sugar. We call it adenosine. And it's triphosphate because it has 3 phosphate on it. Tri, right? Tri is 3, like tricycle has 3 wheels. Triangle has 3 sides. Triphosphate has 3 phosphate on it. So this is ATP, and it's the fully charged version. Okay, this is it's like a fully charged battery. You can use the battery for whatever needs that you need. Okay, like use it for your flashlight, use it for your radio, whatever, right? Okay, so once you use it, right, a phosphate will come off, okay, and then now it becomes only 2 phosphate. So we call it adenosine diphosphate, only 2. Okay, triphosphate is 3, diphosphate is 2. You probably learned this in chemistry, right? Like the tri and the di with the naming stuff. Right? And then more importantly, you release the energy and then you can use this energy for something else. Okay. So in essence, it looks like this. We have ATP, which we generate from catabolic reactions, okay, such as cellular respiration. Then that stores it in the cell. When you need to use it, you add a water molecule and then you release the energy. You can use that energy for various cellular activities, such as powering anabolic reactions for building things, right? But once you use that ATP, it becomes uncharged. It turns into adenosine diphosphate and a free phosphate. In order to recharge it, you need to do more catabolic reaction. Okay, so catabolic reaction will convert this back into ATP, and then you can use it again. So this just keeps on cycling between the 2 forms in the body. As long as we supply energy by eating, we will be able to regenerate ATP, and then we can use the ATP to power different things within the cell. Just one last slide here. What kind of things are we talking about? Right? So, example, mechanical work. If we are talking about a cell that has flagella, right? To move those flagella, you need ATP. To move anything in the body, you need ATP. And moving your muscle, that requires ATP, right? Beating of the heart, right? To talk, right? All those things, movements, we call that mechanical work. Okay, physical movement. Right, so like when you move the hand, right, the muscle cell themselves have to move. That requires ATP, right? And if a body part moves a lot, then you need a lot of ATP, which means you need a lot of mitochondria, right? That was one of the questions we did earlier today. Number 2 is transport work, right? To move molecules across the cell membrane, sometimes that requires ATP. I'll tell you next class what those instances are. And finally, chemical work, right? Like enzymatic work that requires energy. Enzyme lowers the activation energy, but nothing is free, right? Something else has to be supplying that energy. Okay. And in this case, it's the ATP. Right? Anabolic reaction. That's another example of chemical. And so you can use the ATP to do all these things, but afterwards it will turn into ADP and P. And in order to recharge it, we need to do more catabolic reaction. Okay, let me just take this question up with you and then I'll let you go. Catabolic reactions, A, consume more energy than they produce. That is not true. They actually consume less energy than they produce, which is why catabolic reaction ends up giving off energy, right? Those energies then can be used to charge up ATP. Breaks down polymers into monomers. That's correct. Right? The food molecule gets converted into monomers. Produce excess heat to warm the body. That is true because it's not efficient, right? only occurs in human cells and not other cells. That's not true. All living organisms have to have some kind of catabolic reaction to produce the ATP, right? Because production of energy is a characteristics of living thing. All right. That's it. We will finish lecture 4 next class. If we have time, we might even start on lecture 5, because lecture 5 is like a lot of stuff, okay? In fact, I'll be teaching lecture 5 mostly with notes. I mean, I'll have the PowerPoint slides for you, but then I think it's much better if we write out the things together than for me to go through the slides. So we will see. Okay, prepare to learn that on Wednesday as well. But for now, that's it. Thank you for coming and thank you for your participation. Have a good day and see you all on Wednesday. Bye for now.
Thank you.
Thank
Bye.
you.
Yep, no problem.